This is the second half of Chapter 2. We're going to be covering organic molecules in this section. Organic molecules contain carbon and hydrogen, and sometimes oxygen as well. They always contain the hydrogen and carbon, however. The four macromolecules, the biomolecules or organic molecules that make up life are carbohydrates, proteins, nucleic acids, and lipids. The organic molecules are made up of long chains of carbon atoms, which we call a carbon skeleton, and they're linked by covalent bonds. Below is an example of a picture of that carbon skeleton with the carbon atoms with the hydrogens bonding off of them. We also have different function groups that can bond to this carbon skeleton. They form additional covalent bonds with the hydrogen or the oxygen atoms. Less commonly, we have those bonds with nitrogen, phosphorus, sulfur, iron, and additional less common ones. They are soluble in water, and they are highly diverse, but certain groups of atoms show up over and over again. These organic compounds are large molecules of many repeating monomers. The monomers are joined by removing a water molecule, which is a condensation reaction, dehydration, synthesis. If we want to reverse this process and break that large organic compound down back into the monomers, we put it through a process called hydrolysis, which is the addition of water and breaking of these polymers into smaller units. So these functional groups that we talked about on the previous slide, these are particular groups of atoms that greatly influence the chemical and physical properties of any molecule that they're a part of. They have predictable properties. They include acid, bases, the solubility, and bonding. Four common functional groups are amino group, which acts as a base and accepts the hydrogen depending on the pH and can form bonds with other molecules. They are, an example of this is an amino acid. We have a carboxyl group, which acts as an acid. It releases the hydrogen atom to become the RCOO negative, which are found in fatty acids and amino acids as well. The hydroxyl group, which may link molecules through dehydration synthesis. Hydrogen bonding between the hydroxyl groups and water molecules affect its solubility. Examples are carbohydrates, fatty acids, amino acids, and alcohols. And we have our phosphate group, which may link to other molecules to form larger structures, and they also may store energy. Examples of these are phospholipids, nucleic acids, and high-energy compounds. So these are just four examples of the functional groups. There are many, many, many more. We will only kind of focus on these four groups, but just be aware that these are not the only four, and there are so many different functional groups. So first we're going to focus on carbohydrates. Carbohydrates have a carbon-hydrogen-oxygen ratio in a 1:2 ratio. Carbohydrates are found in sugars and starches, and they typically make up approximately half of the American diet. Carbohydrates are a wonderful energy source, and this is the most important role that carbohydrates perform for us. They are catabolized and not stored for later use. Carbohydrates are hydrophilic, which means that they love water. They are easily dissolved in water. The monomers that make up carbohydrates are called monosaccharides. An example of this monosaccharide is glucose and fructose, which are isomers of each other. So the glucose and fructose, like I said, they are isomers of each other. They have the same chemical formula, but the location of the CH2O is in a different location. So arranging these atoms is structurally different. So the picture above in the right-hand corner shows glucose and fructose and shows how that CH2O2 is bonded in a different fashion, therefore giving it different properties and different functions within the body. The body recognizes this structure and understands that it is, even though it is the same chemical components, it is structurally different, and therefore the body utilizes it differently. Then we have the polymers, which are polysaccharides such as starches, cellulose, and glycogen. So the diagram down below are the carbohydrates in the body. We have our structural classes, our monosaccharides, our disaccharides, which is two monosaccharides joined together, and our polysaccharides, which is multiple monosaccharides joined together. The monosaccharides, like we said before, our examples are glucose and fructose. Our disaccharides are sucrose, lactulose, and mallose. And then our polysaccharides are glycogen. So how do we get those polymers from those monomers? How do we build them together? This occurs through dehydration synthesis. So dehydration synthesis is if two hydrogens and an oxygen are removed from a molecule, the octet rule will not be satisfied unless the two atoms covalently bond. So what process could be used to break down large polymers such as glycogen into monomers of glucose? So we're doing the opposite of that dehydration synthesis. We're trying to break down that large polymer that we formed by removing that hydrogen and oxygen. This is called hydrolysis. Remember from earlier when we talked about hydrolysis, hydrolysis is the addition of water. So dehydration synthesis, we removed water, the hydrogen and the oxygen. And now hydrolysis, we're adding the hydrogens and the oxygen back in to break that polymer down into the monomers. Break that polymer down into the monomer. So it reverses the steps of dehydration synthesis. The molecule is broken down by the addition of water. And there's an example of the hydrolysis equation. So polysaccharides are polymers, obviously, of the monosaccharides. They are very complex. They have a repeating dehydration synthesis reactions to create that long chain of those monomers. The addition of the monosaccharides or the disaccharides create the polysaccharides. Excuse me. Starches are formed from glucose, which are broken down in the digestive tract and create our energy sources. So by consuming polysaccharides, we have an energy storage because it takes time to break those starches and the glycogen down in our body, therefore storing that energy for a little bit longer. So the glucagon breaks down the glycogen, which creates the glucose, and this is why we have insulin. Insulin allows the blood glucose to enter the cells, which produces energy. The glycogen, which is an animal starch, is a way to temporarily store our glucose. The muscle cells make and store glycogen. When glucose demand is high, the glycogen is broken down. When glucose need is low, the glycogen absorbs the glucose from the bloodstream and builds the glycogen stores back up. This is not really a long-term energy source. It is, used up pretty rapidly. So if we think of an athlete or a marathon runner, while they're performing, say in a marathon, that's why they use those glucose chews, because it gives them a temporary quick fix because they're using up all of their glycogen storage and they're needing to replace it pretty quickly to be able to finish whatever activity that they're doing. Okay, so what is an example of a monosaccharide? We have glucose and fructose are examples of those monosaccharides. Fructose is a blank of glucose. Now they're both monosaccharides. How are they in relation to each other? It's an isomer. They are isomers of each other. How are complex molecules formed? Are they formed by hydrolysis, decomposition, dehydration synthesis, or catabolism? Dehydration synthesis. So remember, we are removing the water from those monomers to create those disaccharides or those polysaccharides. So we're removing that water out. Hydrolysis is the opposite. We're adding the water back in and breaking those bonds. we've not talked about decomposition or catabolism, so focus on the hydrolysis and that dehydration synthesis. Okay, so now we're going to go on to lipids. Lipids are a little bit different than the carbohydrates, whereas carbohydrates had that 1-to-1 ratio of the hydrogen, carbon, and oxygen. Lipids have a carbon to hydrogen 1-2 ratio, and they have very little oxygen in there. These include fats, oils, and waxes. They are insoluble in water. They are hydrophobic. So carbohydrates were hydrophilic, which is water-loving, and lipids are hydrophobic, water-fearing. They are a very diverse group. They also include phospholipids and triglycerides and steroids. They have many different functions, such as storing energy, their structure, regulation of body temperature, buoyancy, cushioning, insulation. They have special transport mechanisms that carry them into the bloodstream. They are a better source of stored energy than those carbohydrates. It is 2 times more energy than the carbohydrates. The cell membrane also has lipids to help maintain its structure. We have our sex hormones and our chemical messengers. There's 2 main components of the lipids. We have fatty acids, and we have within the fatty acids we have unsaturated and saturated fatty acids. If you look at the picture over there, you can see the saturated, every bond is taken up. We have carbons bonding to hydrogens, and we're using all possible bonding. Whereas the unsaturated, you see that double covalent bond in there between those 2 carbons in the middle. So it's unsaturated. We don't have every bond taken care of. So that's the difference between saturated and unsaturated. So glycerol is a molecule that fatty acids can attach to. These fatty acids cannot attach with dehydration synthesis because there's no oxygen there. There's very little oxygen, so there's very little water in lipids. And remember, lipids don't like water. So we can't use that to create more bonds. We must use that glycerol. This creates a glyceride. This word might be somewhat familiar. Think of the word triglyceride. So this is where triglycerides come from. We create multiple bonding between those glycerides. So triglycerides are something that we measure in our cholesterol panel when we draw blood, and it obviously is the fat in our blood. So a key functional group for the lipids are the carboxyl group. You can see with that picture on the right-hand side there. So that carboxyl group is key for bonding for these lipids as well. So we have our fatty acids. Fatty acids have a long carbon chain with hydrogen and a carboxyl group. The longer the hydrocarbon tail, the lower the solubility of the molecule. So this carbon tail is hydrophobic. It does not like water. For this reason, it has very limited solubility in water. So we go back to the statement above. The longer the hydrocarbon tail, the lower the solubility of the molecule. So the longer of that tail, the less likely it's going to dissolve in water, or it's going to be really hard to dissolve in water because that tail is so hydrophobic. And it's just gonna, so if you think of you're pouring oil in a pot of water, that oil sits on top of the water, or if you start to try to boil that water, that oil is going to kind of clump together. It's not going to dissolve in that water. It's always going to be visible in the water. So that's this concept here. So then we have our saturated versus our unsaturated. In saturated, each carbon atom in the tail has 4 single covalent bonds. In the unsaturated, 1 or more single covalent bond has been replaced with a double covalent bond. 1 carbon atom only binds to 1 hydrogen atom. This changes the shape of the hydrocarbon tail and how the fatty acid is metabolized, because a double bond is harder to break than a single bond. So that's going to affect how it's metabolized and how long it takes to metabolize in the body. Okay, so in hydrolysis of a triglyceride, what are the reactants and what are the products? Is it A, the reactants are triglycerides and water, products are glycerol and 3 fatty acids? Is it B, reactants are the diglyceride and water, the products are glycerol and 2 fatty acids? Or is it C, the monoglyceride and water are the reactants, and the products are glycerol and 1 fatty acid? So to help you figure out this question, kind of break down the word triglyceride. So tri means 3. So that may help you right there. And then glyceride obviously is a product of the glycerol. So use that information to try to figure out your answer. So the answer is A. So the reactants are the triglycerides and the water, which breaks down into the glycerol and 3 fatty acids. And there's an example of what that reaction would be written out. Okay, so we have a lot of different types of lipids in the body. There are fatty acids, there are glycerides, which we have covered already. Then in AP2, you're going to be covering there's steroids, those phospholipids, those glycolipids, the eicosanols. So there's so many different things that are in lipids in the body. All right, so now we have our phospholipids and our cholesterol. So this is essential to the cell membrane, these phospholipids and cholesterol. The phospholipids have a hydrophilic head. So remember, hydrophilic means they like water. And they have a hydrophobic fatty acid tail. So those tails don't like water. So the head likes the water and the tails don't like the water. So we're going to go a little more in depth into those phospholipids in chapter 3. We're just going to kind of touch base right now on those. And then we have our cholesterol, which are needed to maintain their plasma membranes and for cell growth and division. So there's always a big controversy about cholesterol and diet and cholesterol medications, statins. Are statins good for you? Are they bad for you? Is cholesterol good for you? Is cholesterol bad for you? The short answer is that everything in moderation is good for life. It doesn't matter what it is. So that with that being said, cholesterol, we need cholesterol in our diet, we need cholesterol in our body, but we don't need an abundance or an excessive amount of it. Then we have our steroids, which are a 4-carbon ring, which include our estrogen and our testosterone. The phospholipids, the cholesterol, and the glycolipids are structurally related. They are structural lipids because of their roles in forming and maintaining membrane structures. The long carbon chains are hydrophobic, and the opposite ends are hydrophilic, which is a non-lipid head. In the water, molecules clump together in mesocell. So this is a little picture right there of that mesocell and how those hydrophilic heads are on the outside because they like the water, and those tails are inside that little tiny circle because they don't like the water. Most foods contain lipids. When a food is broken down in the digestive tract, this mesocell forms. Okay, so a quick question. Which type of organic molecule forms the bilayer of the cell membrane? Is it DNA, the phospholipids, the steroids, or the amino acids? It's the phospholipids. All right, so now we're going to move on to proteins. Proteins are our most abundant organic molecule in the body. They're made up of long chains of amino acids, and they're held together by covalent bonds. The monomer is the amino acid, and they combine to form these polymers. There are 20 different amino acids that are the building blocks of life. They function as support, storage, transportation, signaling, defense, movement, and catalysts in our body. The carbon, hydrogen, oxygen, and nitrogen are present in all amino acids. All proteins have a central carbon, a hydrogen, an amino acid, a carboxyl group, and an R group. And down there on the bottom right-hand side is an example of what that would look like. Proteins make up approximately 50% of our cellular dry weight, so they are a huge component in our body. So how do we form a protein? The protein is made up of 3 functional groups: the carboxyl group, the amino acid group, and the R group. The carboxyl group acts as an acid. It releases a hydrogen ion to become the carboxyl ion, and the amino acid acts as a base and accepts that hydrogen ion to become an amino ion. And then the R group is what makes each protein special or unique. This R group can be polar, it can be nonpolar, or it can be electrically or charged or even uncharged. So how do we form multiple protein chains into polypeptides? We do this through dehydration synthesis. It forms covalent bonds between the amino acids, which are called the peptide bonds. So peptide bonds are just another fancy way of saying a covalent bond for a protein. And to your right side there is an example of that reaction. So the polypeptides become proteins at 100 plus amino acids. We have 4 different levels of structural complexity. We have primary, secondary, tertiary, and quaternary. So the primary is a sequence of amino acids bonded together in a linear chain. The secondary formation, the bonds between the atoms at different parts of polypeptide chain, and the hydrogen bonding can create a spiral chain or a pleated sheet. That spiral chain is called an alpha helix, and that pleated sheet is called a beta sheet. The helix is the most common form of a secondary polypeptide structure. The tertiary is a folding of that polypeptide chain into a 3D shape, and it primarily interacts with the polypeptide chain and surrounding water molecules in an R group, but not so much. Then the quaternary structure, those polypeptides come together to form a large complex. The shape of the protein determines its function. If it's altered, it will likely not function. This is called denaturization. Denaturization occurs when the temperature rises, when changes in the protein shape and enzyme function deteriorates. This happens to our organs and our organ systems when our body temperature rises and sustains above 110 Fahrenheit. So denaturization is basically the death of your cells, because the cells can't maintain their homeostasis at 110 degrees Fahrenheit. Any other environmental factors can also contribute. If the body's too acidic, if the body's too basic, all these things also could denaturization of that polypeptide chain or structure and cause it to die as well. This occurs a lot in sepsis. That's why it's really important to get the infection under control, get the body back to homeostasis, because in sepsis there's so many different things that can happen. Elevation of temperature, changing of the pH of the blood, may be more acidic or more basic because the body's trying to fight that infection. Also, we find this also with respiratory failure. The body can either be basic or acidic based upon what the oxygen level of the body is doing. So there's so many different ways that the body can go one way or the other, and we're trying to overcorrect ourselves, and then those, like I said, those cells start to die. So these are the pictures of those examples of those polypeptide structures. That primary structure there, that linear chain. The secondary structure, we have that alpha helix, that spiraled looking structure, and then that beta sheet, that pleated beta sheet. Our tertiary structure, this is an example of a hemoglobin. So this is a single heme unit, and it creates that 3D structure. You see that heme unit right there in the middle, kind of looks like a disc with a ball in the center of it. And then lastly, we have that quaternary structure. So the first example is that hemoglobin molecule. We took 4 of those polypeptide subunits of the hemoglobin, and we combined them together to create this big, looks like almost like a big jumble of ribbons or string all kind of wound up together. And in comparison, the collagen fiber, it looks completely different because the R group attaching to that polypeptide chain is different from the hemoglobin. So the collagen looks more linear. It looks like a braided rope because the purpose of collagen versus the purpose of hemoglobin are very different in the body. Collagen is found in our skin and our vessels and various other places to help with the stretchiness and reactivity of the body to different environmental factors, whereas the hemoglobin, its main responsibility is to transport nutrients and oxygen throughout our entire body. So that R group's going to determine what is the purpose of this polypeptide and what should this polypeptide look like based upon its job. Okay, so a quick question. The building blocks of proteins are amino acids, nucleotides, fatty acids, or monosaccharides. They are amino acids. So remember, those proteins are made up of amino acids, and then we combined all those proteins together to form those big polypeptides. All right, so now we're going to move on to enzymes. Enzymes are a group of proteins that catalyze chemical reactions. They are catalysts that are compounds that speed up a chemical reaction. So they lower the activation energy required to create a chemical reaction. They are not altered by the reactions they participate in. They're reusable and will continue the reaction until equilibrium is reached. So they're not, for example, it's not like gas. Once we use up the amount of gas we have in our car, we got to put more gas in there. That's not the scenario with these enzymes. They are an endless supply of energy, and we use them as long as we need them to create that equilibrium homeostasis in the body. But before a reaction can occur, enough energy must be present to excite these reactants. So once we are at equilibrium, that switch is turned off. Even though those enzymes are still there, they're not going to keep creating that reaction. This is where that excitation, enzyme reaction, those voltage gated channel, the sodium potassium pump, this is where all these come into play to create that imbalance in energy to get those enzymes excited and get them reacting. So this is the activation energy. The reactants can be exergonic, which are more common, and they release energy, which makes heat that controls our body temperature. Or we can have endergonic, which are less common. These guys store energy, have absorption of energy. This is an example of ATP synthesis. Life requires these metabolic pathways. So this is an example of a metabolic pathway. We have a substance A, and we have that reactant, that enzyme, and substance B. So there's another enzyme too that creates from B to C, and then C will react with enzyme 3 to create D, and so on and so forth. So the reaction just keeps going and going and going. These enzymes that create these reactions are highly specific, so they will only react with certain types of products. So how do these enzymes carry out these reactions? Their shape is essential to function. Once I said before, the enzymes are highly specific to each product. So we have an activation site, which is the region on the enzyme where the substrate binds. So the first picture on the top there shows our little blue enzyme there, and then we have that little activation site. We have those substrates floating around and hoping that they are the correct fit to that activation site. Once we find that substrate, which is a reactant in a chemical reaction that fits that activation site, it can bind. So the product is that end result of that chemical reaction. So once that substrate binds to that activation site, we're going to create that new product. So remember, enzymes are highly specific. They only bind to a certain enzyme and certain active site. They facilitate a single type of reaction for a particular substrate. They are proteins coded by our genes for your DNA. So we have a lock and key model. That's what this picture is alluding to, that specific substrate only fits in that active site. So it only fits in that one key, that substrate only fits into a specific lock, that active site. So the induced fit model, which is the example in the picture over there, the science requires us to modify a hypothesis based on this evidence. As we gather more data, we often must adjust our initial hypothesis. This is a scientific thinking. So this is the lock and key method and the steps that we go through for that lock and key. So the substrate binds at an active site, which is our lock and key. The structure of the enzyme determines the shape of the active site. Specificity is determined by the ability of the active site to bind only with substrates with a certain shape and charge. Anything that changes the shape of an enzyme can turn it on or off. When the substrate binds, it temporarily changes the enzyme's shape, and this creates the reaction, and the product is then formed. The product then detaches from the active site, and the process is repeated until equilibrium is reached. So what factors are going to influence the rate of an enzymatic reaction? Temperature and pH and salinity are all going to affect that rate of that reaction. Warmer temperatures speed up reactions. Molecules are moving faster and more likely to come in contact with enzymes. So you can think of a hurricane. Hurricanes like warmer water. In the winter months, our waters are colder. Hurricanes are not going to develop because they can't form, because it doesn't have that increased energy from that warm water. But temperature, pH, salinity out of the normal range can also lead to denaturalization. So we have that fine line of what is too warm, what is too cold, what is if the pH is too high, too low. So it needs to stay at that perfect level to create that enzyme reaction. The concentration of the enzyme in our substrate obviously can influence the rate. If lots of substrate but only a few enzyme molecules, it will be slower than if it were lots of enzymes to simultaneously facilitate the chemical reaction. So, for example, what it's saying is I have 3 enzymes, but I have 100 substrates. I can only bind however many substrates that enzyme allows me. So I can only bind, let's say, two substrates per enzyme. So it doesn't matter that I have 100 substrates sitting there. I can only bind those 2 substrates to each enzyme at a time. The reaction occurs. The reaction is the same for each substrate. So I can only produce the same amount at the same time. So what do you think happens to the rate of the reaction if we increase the substrate concentration? I just answered that question for you. It doesn't matter if we have 100 substrate, 1,000 substrate, or 10 substrate. If I only have 10 enzymes, I'm only going to get the same reaction for those 10 enzymes. If I have more enzymes, my reaction is going to go faster because I have more active binding sites. But if I don't have enough of those binding sites, doesn't matter how many substrate I have, my rate of my reaction is going to stay exactly the same. So the answer, initially the rate increases, but when the molecules are saturated with substrate, the reaction rate levels off, which is our saturation limit. So we have ATP, which is our high energy compounds. This enzymatic reaction may require energy. That's why we need the ATP. In the cell, energy mostly comes from ATP, which is produced in the mitochondria. The ATP contains covalent bonds that have a lot of potential stored energy that can be released as kinetic energy. This is the energy that does work. It provides energy to power many vital functions. For example, muscle contraction, which is the breakdown of proteins, carbohydrates, and lipids. The adenosine triphosphate is formed by combining adenosine diphosphate with a phosphate, which we get ATP. So we have the adenosine diphosphate plus energy plus that phosphate. We have that enzymatic reaction of that ATPase. Then we get that ATP and we get that H2O because we perform this reaction by dehydration synthesis. So the ADP to ATP is reversible. Energy is stored with the formation of ATP, then released when broken down into ADP. The hydraulic breakdown to ATP requires ATPase. So both ways it requires that ATPase, that enzyme, to create that reaction. So we're going from ADP to ATP. We're creating a dehydration synthesis reaction. And if we're reversing that ATP back to ADP, we're doing a hydrolysis reaction. All right, so now we have nucleic acids. Nucleic acids consist of DNA and RNA. They are a combination of carbon, hydrogen, oxygen, nitrogen, and phosphorus. They store and transfer information, which is their primary job. The polymer is made up of 1 to 2 long chains of subunits that are formed by dehydration synthesis. The monomer of nucleic acids is a nucleotide, which is an individual subunit of the nucleic acid. Then there's 2 different types of nucleic acids, the DNA and the RNA. DNA is a double helix with deoxyribose. It also has adenine, thymine, guanine, and cytosine. RNA is a single helix with ribose, and it has adenine, uracil, cytosine, and guanine. So the difference between DNA and RNA is that deoxyribose and ribose, but also that uracil and that thymine. The nucleotide has a phosphate group. It's a 5-carbon sugar with a deoxyribose or the ribose, a nitrogenous base, which is a purine or a pyrimidine. The phosphate and sugar can be joined together by dehydration synthesis. This makes the longer chains in the backbone of that nucleic acid. The sequence of the nitrogenous bases can carry the information for protein synthesis. So those purine bases are adenine and guanine, and the pyrimidine bases are cytosine, thymine, and uracil. So those nitrogen bases, which I just said, the purines are adenine and guanine, which are found in both DNA and RNA. The pyrimidines, which is cytosine, thymine, and uracil. Cytosine is found in both DNA and RNA. Thymine is only in the DNA, and uracil is only in RNA. Purines always bind to the pyrimidines. Never do 2 purines bind together or 2 pyrimidines bind together. The purines have a double ring, as you can see with that picture to the right-hand side, and the pyrimidines have a single ring. So DNA and RNA. DNA is 2 complementary strands creating that double helix. The hydrogen bonds hold strands together but are weak, so they can be broken and unzipped and re-zipped for copying, which is important for that RNA production. The complementary base pairs are adenine and thymine, and cytosine and guanine, which is that purine-pyrimidine combination of bonding. The RNA is a single chain of nucleotides. The shape and function depends on the order or the nucleotides and interactions. There are 3 different types of RNA based upon what their job is. We have messenger RNA, which that copy of that gene, it goes to the ribosome. The transfer RNA matches the amino acid to the messenger RNA code, and the ribosomal RNA makes up the ribosome, which is the site of the protein synthesis. So what are the complementary base pairing rules for DNA? Cytosine pairs with cytosine, adenine pairs with adenine, thymine pairs with thymine, guanine pairs with guanine, adenine pairs with cytosine, guanine pairs with thymine, guanine pairs with thymine, cytosine pairs with adenine, adenine pairs with thymine, and guanine pairs with cytosine. Adenine pairs with thymine, and guanine pairs with cytosine. Okay, so this last slide is a comparison of DNA to RNA. It just shows the difference between the two, the sugar base, which is the deoxyribose versus the ribose, the nitrogen bases, which we've covered, the adenine, guanine, cytosine, thymine are found in DNA, and then the adenine, guanine, cytosine, and uracil are found in RNA. The number of nucleotides is in a typical molecule. There's always more than 45 million in DNA, and the RNA varies from fewer than 100 to about 50,000. The molecular shape: paired strands collide in a double helix, whereas the RNA varies with the hydrogen bonding along the length of the strand of each of the three main types of messenger RNA, transverse RNA, and ribosomal RNA. And then the function of DNA is to store that genetic information that controls protein synthesis, and then the RNA performs protein synthesis as directed by the DNA. So this concludes Chapter 2. We're going to be moving on to Chapter 3 next. Make sure you are completing those homework and quiz assignments and doing any preparation for our upcoming test after we finish Chapter 3.